Okay, thank you. So first of all, thank you to all the people named in organising and those that David perhaps forgot to name as well. And thanks very much to Nimbus for funding this. It's a great honour to be speaking and to be speaking first. And thank you to David for suggesting a title. It's one of the few times I've been in a presentation where the title was given for me, so thank you for that, David. So yeah, I'm really looking forward to the next couple of days. So David tasked me with giving a kind of general overview to sort of set the scene and think about some of the perspectives and challenges. And when thinking about this, I thought, well, it'd be good to look forwards, backwards, and where we are currently, and then to draw on some of the things that I think are challenges in my own work and the work of others. But before I get started, I normally have the acknowledgements at the end, but really they need to come at the beginning. For the last year or so, with Dick in the audience and Jens Krause, we've been putting together an edited book on animal social networks. Some of the authors are here, and I've been editing section 2 on patterns and processes, and so a lot of the ideas I'm going to talk about today have come from this work and come from reading these chapters and editing these chapters. So this book, we hope, will be out sometime in 2015, early 2015. We're just putting the final touches to it at the moment. So these people need a lot of credit for what I'm going to talk about today. So the aim, I thought, would be good to give a kind of general overview, given that this is an interdisciplinary audience. I'm not sure how familiar everyone is with the type of work that we've been doing in behavioural ecology and the sort of animal behaviour. So get an idea of what we're talking about when we talk about animal social network. How did we get here? How did I get here? I thought that would be quite interesting to think about, how did I get interested in networks and what are some of the lessons I've learned along the way? But then thinking about the future, and I think this is really exciting, and certainly from reading through the chapters in the book, there's a great excitement in what can be done. But there are still some challenges to get there, and so I'll highlight a number of areas that I think really we can make some really big progress by applying a network perspective to behavioural ecology. But then there are some issues, and we heard very, very rightly comparing networks is a challenge, and I think that's a challenge that would be good to talk about today. So the kind of network thinking or sociogram has been around for a long time. So right back in the 1950s, people were starting to draw networks, but calling them sociograms, and even having weighted edges on these networks. But the statistical analysis of that didn't come until later. And it wasn't just in primatology, so this moved over into other areas. So here we can see sociograms here of some dolphins in the 90s. So I thought it'd be just kind of plot what's happened in terms of papers if I just do a quick Web of Science search. And this doesn't capture all the papers, but I just use the search term social network and animal. And around 2003, 2004, 2005, we started to get the kind of quantitative analysis, boring methods from other disciplines. And we saw this rapid sort of exponential increase in the number of papers coming out with the term animal and social network. And these papers are well cited, so we can see the things that at least we're interested in terms of average citations per paper. It all looks very healthy. But it's difficult to know what's going to happen over the next few years, but it does seem to have sort of leveled off a little bit, and I think that this has the potential to keep going a lot higher and to pick back up that steep curve, and I'm hoping one of the things that we can do is to help encourage that through the next couple of days. Again, for those of you in the audience who are not necessarily biologists, this is very taxonomically broad. So, going from insects, here we can see some beetles, ants, fish, birds, reptiles, marine mammals, primates, have all been considered from a network's perspective, whereby, in this case, individuals and nodes and the edges are either a measure of their social interaction, where one animal has groomed another, or one animal has been aggressive towards another, or inferred through patterns of association. I'll talk a little bit about that in a moment. So how did I sort of get interested in this as a method? So I way back 10, 12 years ago, started my PhD and went out to Trinidad, having read how wonderful the guppy, so this is a small freshwater fish that lives on the island of Trinidad, is sexually dimorphic. We can see females there, the big brown ones, and males, the small ones with all the colour. How they have these incredible social structures. So doing simple, and I've done some myself in the lab, simple experiments in the fish tank where you can confine fish and give them a choice between going to one side or going to the other side. They recognize familiars. They prefer to associate with familiars. They have individual recognition. They remember the behavior of previous partners in terms of their competitive ability, their behavior during predator inspection. So I expected to go down to the river and see this very organized society with animals, fish swimming around with their friends. And I was very stressed when I got there because we'll see if this works. But this is kind of, I don't know if you can see these from the back, the video's not that good, but here we can see some guppies kind of milling around in the shallow water of a stream in Trinidad, and they're foraging on rocks here. The fish are individually marked, so we can pick them up and ID the fish. And yeah, they do shoal and they do school when they're out in the deeper water, but certainly in the shallows, they're spending a great deal of time on their own. When they are meeting individuals, it's relatively brief. There's some mating, some courtship going on there, a male displaying to the female at the top, but it's extremely dynamic. So associations are lasting for a few seconds, individuals are moving between groups. I thought all of this from the laboratory was simply an artifact of housing fish in tanks. And there's some predation going on. We'll see. This is a rivulus coming in here and trying to eat the guppies, and they're coming together, and then they are starting to have greater cohesion. So really, I came to a networks approach with trying to capture this very complex and very dynamic system with animals joining and leaving groups on a regular basis, and really wanting to ask, well, can fish really maintain, do fish really maintain these stable associations, call them relationships if they're engaged in behavior, under such dynamic fission fusion? And so we really need to get a way of capturing this complexity. So you saw the fish were marked. We were able to tag these fish with these colours that we inject under the dorsal epidermis, and we had to do the appropriate control experiments to show that the mark doesn't affect the behavior of the fish. And fortunately for us, we get to study these fish in these rainforest streams, which is beautiful. And we were defining associations based on animals within spatial proximity, so being together within the same group, and through repeatedly capturing or sampling groups of fish, either with photographs or actually going into the river and catching the group, we can record who is with whom through time and then build up a matrix of associations of fish within the same shoal. And we did this, and we can then use that matrix to draw a network, and we can see then the fish are color-coded by the nodes of males and females. So we see the females tend to be more in the center and the red dots around the outside, and that fits with what we see with fish going on with males moving between groups to mate with females. We were very excited at the time about, wow, we now have a network, we can now measure things on that network, we can borrow from mathematical approaches and approaches to other networks, and we can start measuring things like degree and path length and clustering, and drawing comparisons between what others were doing and our own networks that we're observing. So for example, we're seeing here that the clustering coefficient is higher than you'd expect on a random graph, but the path length is very similar. And we got excited, as many others did, about the idea of a small world. And we plotted other things like the degree distribution, looking at whether or not fish that have a high degree are also associated with others with a high degree. Looking back, a lot of this structure is influenced by how we sample the animals. So actually, it would be difficult to get anything but a small world where we're sampling. And the fact that these animals are living in groups kind of, to some extent, forces them to have a positive degree distribution. So it's great to be enthusiastic and to borrow and to mine the other approaches out there, but we need to be very careful in thinking about how robust they are when applied to our data. So it's not all bad. That wasn't the main question. We were interested in asking about these stable interactions. So a way to visualize this was just to kind of filter this network. So we were looking at, well, who's hanging around together repeatedly, and seeing these clusters of females still there after these are fish that were caught together 3 or more times over a 7-day period, and so there were these clusters of females there. So boring methods in terms of looking at the repeated pairs, so using randomization tests that control for the group structure, we were able to test and show that females occur together more often than we'd expect by chance. So there's repeated interactions between these females. But again, is that really satisfactory? I mean, could this just happen by chance? Could it be that females just happen to be in the same place at the same time, that males are more mobile, they're leaving the pool, so it's kind of forcing this pattern to occur in the data? So one of the things I think we haven't really made enough of, and some systems we can't, is experiments. And one of the things we did quite early was to test this hypothesis by taking our laptop down to the stream, drawing the network or constructing the network, but then pulling fish out of the river and giving them a choice between shoaling with their direct neighbor or a fish of the same size, the same sex, from the same pool, but who they didn't have a direct connection with. So actually experimentally testing the significance of these associations we're inferring through the group composition. And fortunately for us, fish spent significantly more time associating with their observed associate than a control fish, than a matched sex, matched size control fish. So we did find evidence that these stable interactions occur, and this is a nice sort of illustration of how to actually test, not all suitable for all systems, but how to test these things experimentally. And I do think that's really important, and important for how we move forward in terms of testing patterns and testing hypotheses. So what did we learn from this? So associations were differentiated. I think it's easy to borrow measures and borrow techniques, but we need to think carefully whether or not they're really going to give us useful information and are applicable to our system. And it showed us that we can capture the social structure of populations. But the real potential, I think, is in using this to test hypotheses or to develop models of social behavior against which we test our observed data structure. And there's two, at least in the way I've been thinking about this, there's two ways we might go about approaching this. We can think about this going from a top-down perspective. So what are the consequences of population social structure for individuals? Or the other way around, how do individuals shape the social network structure at the community or population level, so sort of from the bottom up? And so just to give you some examples of how we've approached this, here's a study that Safi Darden did as a postdoc and has later gone on to do a lot more work on this topic, looking at sexual conflict and sexual coercion. So in this project, male guppies harass female guppies almost constantly for reproductive opportunities, and they have internal fertilization. So females spend most of their time being harassed. We've considered the kind of costs of harassment in terms of energetic costs. Females are moving away from males, they may get physically damaged, there may be increased risk of predation. Safi was interested in the social cost, or potential social cost. So she housed fish in these groups these are networks of large females, either housed with males or small females, so the males and the small females are not in these graphs. And this is the resulting network accumulated, I think, just over a short time period, kind of 30-minute sampling period with 30-second intervals. So we can see that when these females with males, their network is more fragmented, so we can ask, what's the consequences of that for the individuals? So this is the results of an experiment looking at the development of individual recognition, so the ability of these fish to actually learn and remember and show preference for individuals they were being housed with. This is the treatment group, this is time down here in days, this is 0, so no preference. This is using one of those binary choice shoal tests that we saw in the earlier slide. So this is the females with males, and this is the females with females. So this structure here is actually preventing these females from generating familiarity until later on. When they do generate it, they show a preference for avoiding the fish that they're socially familiar with. These fish, in contrast, when they're housed with the small females, so control, they show preference for, they develop individual recognition quicker and then show an active preference for the females that they have been associating with. So this structure here in the network is influencing the behavior of these individuals. So we can approach it the other way and ask how the individuals shape the population structure. So this is an experiment that we did with guppies, and they're infected by this gyrodactylid, this parasite that's transmitted socially, so it clings onto the side of the fish, and it's transmitted either through direct contact or through contact with substrate in the environment. And so we were interested in thinking, well, what happens when an infection is introduced into a population? How does the population respond to that infection? So we generated groups, or mesocosms, small pools of fish that were infection-free, and then we introduced either an infected fish or a control fish. And we measured a number of properties of the structure of the group. This is just a measure of clustering coefficient here on the y-axis. And so as soon as you introduce a new fish, clustering coefficient drops dramatically, so regardless of the infection status, but it stayed lower for the infected fish after 24 hours than for the uninfected fish. So the group that had uninfected fish, they started to regain or resume that clustering, whereas where the introduced fish had been an infected fish, that clustering still stayed at a much lower level. And we didn't go on to study this over a later time period, but it just illustrates here how we can take the other approach and ask about how individuals are shaping structure at higher levels. So in terms of thinking about the perspectives and the future around 2008, 2009, 2010, there are a number of really reviews and great enthusiasm for embracing this social network approach and for just the potential that it can bring to behavioural ecology and the study of animal behaviour, and the recognition that social structure matters for individuals, populations, and population processes. And today, from being involved in editing this book and from reading a lot in preparation for this meeting, I just wanted to kind of focus on three areas, not to say that the other ones aren't important, but on three areas rather than trying to give a little bit on everything, where I think really we can learn something, where possibly the only way really to learn something is by adopting this approach. And I did have green and red, but then realised that's not good for 1/4 of males in the population. So I changed it to green and blue, and hopefully that doesn't cause the same problems. So just to kind of think, well, what do we know? And then what don't we know? So what are some of these questions that we can go out there and ask when we're armed with our tools? So I picked three: the evolution of cooperation, sexual selection, and collective behaviour. So the evolution of cooperation has really challenged biologists since the time of Darwin. So why would one individual pay a cost for another to receive a benefit? And kin selection obviously does a good job of explaining that when it's between relatives, but what about cooperation between non-genetic relatives? And so there's this average fitness is maximised when all individuals are cooperative, but in a well-mixed population, cooperators are open to exploitation by invading defectors, and thus cooperation is evolutionarily unstable. But we know that populations are not well-mixed. So these early models that assume a well-mixed population, we know that's not the case. We've seen the pictures of the guppies that show that societies or these groups are actually highly structured. And so back in the 90s, Noack and May demonstrated that spatial structure, playing games on a lattice here, actually can lead to stable cooperation. So here generates these lovely kind of 60s carpet patterns with red squares here being cooperators, and you see they're clustering in space. More recently, within the last sort of 6 to 8 years, for a social species, it's kind of unrealistic to think there's just birth and death in these squares. Actually, they're interacting in a more dynamic or a more complex way in a network. And so again, here we can see a study just demonstrating that assortment really is key, and if you have assortment of the network, certain properties of the network in terms of low degree, then you can get stable cooperation. And it turns out that actually updating social partners is important, so social dynamics and the ability to switch between partners, so to generate new links, so moving cooperator here, breaking the link with the defector and forming a new link with one of the defector's associates also appears to be important. What all of these have in common are there are different ways really of getting assortment among cooperators. So these models really tell us that the key to cooperation is assortment, either among kin or among cooperators. So as a biologist rather than a theoretician, I'm interested in those mechanisms. So what mechanisms have been proposed that can drive this? So the non-random distribution of individuals in time and space, so thinking about cooperators being spatially structured, clustered in space and time. And many of us will be familiar with these, so condition-dependent strategies, tit for tat, you scratch my back and I'll scratch yours. Certainly happens in humans. The extent to which it happens in animals has been greatly debated. There are more cognitively much simpler mechanisms, such as generalized reciprocity, help anyone if helped by someone else, and there is a suggestion that this actually happens in animals. Certainly it happens regularly in human societies. And another model which I think is very interesting is partner choice and breaking of those links, so the dynamics of that network, and walk away when encountering non-cooperation. So in theoretical models have shown that that can drive assortment among social ties in networks by cooperation. So we're provided with a number of mechanisms that predicts that can lead to assortment. This has kind of been tested in animals, and there's a great deal of debate. As far as I'm aware, there's only one study that really has done this in great detail, and we know very little about how individuals, the rules that individuals make when breaking and forming new links in networks. So some of the blue things then. Here we can just see a figure here that just shows actually there's variation among individuals in how cooperative they are, so that's something we can measure. This is their cooperative score, and this is just something particular for guppies, and that's the rank, and we can see that there's consistent differences among individuals. But we know virtually nothing about how these individuals are distributed in space and time in real populations. So what's the spatial distribution of cooperators? And I think that's really exciting. And how does space interact with social interaction? So how does the spatial distribution of animals drive the assortment that we're measuring in the networks? So do individuals use these simple condition-dependent strategies? So the idea of generalized reciprocity, I think, is something that would be really exciting to test in the field. So how do individuals make decisions about cooperating in real networks where they're faced with multiple choices of individuals? It's not just dyadic. And what type of rules do individuals use for forming and breaking links in networks? So is there evidence that they're using kind of simple strategies like a walk-away rule? I encounter a defector, I just move to a new group. In theory, that can drive assortment. Very simple, doesn't require complex cognitive ability, but again, we don't know what's going on. So to switch to another topic now, sexual selection. A major driving force for Darwinian evolution. It can lead to speciation, to local extinction. So for evolutionary biology, measuring sexual selection and looking at factors that influence the strength of selection has been really important. But a kind of theme coming through here when we think about cooperation, traditional selection analysis assumes competitive and selection environments are homogeneous. So we're thinking of kind of a well-mixed population again, which we know simply isn't the case. There have been these kind of multi-level selection approaches to incorporate population structure, but it's kind of difficult to define a priori what that structure is. What should those communities be in advance of doing the analysis? So another way to think about this is a sexual network. And again, these aren't my ideas. This is work by Grant and Tommaso in Oxford, at least in thinking of sexual networks. But here we can see an empirical study. I'll come on to their work in a second. But here we can see an empirical study that has considered a sexual network. So sexual selection is driven by social competition. So social network structures like to have a profound effect on the strength of selection. So here's a really classic example of that, looking at the house finch and incorporating community structure into the measure of sexual selection and standardizing individual traits, not at the population level, but at the level at which they're interacting within their social community. And this showed that individuals are capable of influencing the selection regime by modifying their social environment. So dull males could look relatively more attractive by associating with even duller males. So it's not uniform at population level, there's structure. But this considers a community level. What about structure within communities? We know that within a community, it's not homogeneous, that there's social structure within a community, and that there are edges between communities that might be important as well. So now moving on to the work by Grant and Tom and Dick, who's in the audience as well. Kind of captured that by thinking of a sexual network whereby you can draw a network based on males in competition with other males. So in this figure here, we can look at a male's competitive environment by describing that as a network, and in this here it's a network relating to sperm transfer, so ejaculate transfer, and this is just showing the network for male A here, who mates with female B and female A, who's in competition in female B with male D and male E. So we can have this kind of egocentric approach where we can describe then who's in competition with whom and capture that competitive environment not on a community level but on a local level, actually measured at the individual. And so Grant and Tom and Dick and Jens that were involved in this running simulations suggested that this approach can capture sexual selection far more accurately than traditional measures, especially when there are particulars about the mating system, which I don't have time to go into. But there are still some questions. This is a kind of theoretical study. What determines the competitive landscape, and how is this influenced by female behavior? How does this, what are the mechanisms that drive this structure? How much do individuals move in the population? Of what scale does this happen? The female mates with the male, how far does she move? How connected are these networks in space? I think that's a really exciting area to explore in the future. So one thing that's been really, so moving topic again, one area of research that's been really exploded recently over the last 10 years has been the study of collective dynamics and collective movement in animal societies. So here we can see some sardines here schooling very tightly, creating these amazing collective phenomena. We could be watching starlings flying or birds flocking or insects swarming. And understanding the mechanisms that drive this structure has been a really exciting area of research. And simple model, so here's a model by Ian Cousin, and I apologize, this is quite small, it will be familiar to many of you in the audience, demonstrated that using very local information, so just having a zone of repulsion, a zone of orientation, and a zone of attraction, you can get these kind of complex patterns at population level by individuals just using very local and simple rules. And again, this is just a video I found online, but we can visualize how those rules kind of play out when they're played in an agent-based simulation. So here we can see a predatory fish kind of attacking a school, and we're getting exactly the same patterns that we'd see in the fish in the video in terms of vacuoles and flash expansion, just with individuals paying attention to these local rules at a very local level, orientating, attraction and repulsion. And studies on real animals have shown this actually happens. So here's some work on starlings that's demonstrated individuals really do pay attention to local neighbours, and it's around, the magic number seems to be somewhere between 6 and 7, by taking still shots of these flocks just before roost, actually looking at how individual trajectories are changing based on their local neighbours. So we can get these complex patterns emerging from local interactions, but this assumes that all associations or individuals are interchangeable. So these agents don't have any social preferences. You could switch any agent with any other agent. And that may be true possibly when we look at this kind of sardine school, but certainly for some of the other animals we're interested in, we know their structure, we know their preferences, even in something like the guppies. So how does this play out? What's the consequence of this? So Nikolai Bode and Dan Franks had a review paper where they kind of summarized this and term this a communication network whereby, and I think that's slightly misleading, so I call it an interaction network, whereby individual 1 at this point in time can interact with individuals 3, 2, and 4, and those preferences are not just going to be based on how far those individuals are away, but they're going to be influenced by the social network, by their existing preferences to associate and assort with those individuals. And we've thought about animals, but if a fire alarm was to go off, we'd all have to run out of here or walk out of here in an orderly fashion without collecting our bags. But if I had my kids here, I'm going to want to pull them along with me. I'm not just going to act as an independent agent. So modeling human evacuation dynamics has shown that social structure can really affect how individuals move through space and those preferences. So theory predicts this is important, but really we know there's no empirical data here, and I think this is a really exciting area for future work. So how do social networks determine patterns of group movement, fission and fusion in real-world populations? But what's the feedback there between also being involved in collective behavior, and then how does that influence an individual's social network? So there are just some topics that I think are really ripe and exciting for future work. And what are some of the challenges? So in terms of thinking about cooperation, experimental manipulation, actually manipulating cooperation outside of a kind of dyadic context in a real-world network is difficult but possible. We've heard something about comparing networks already. I think there's great potential there if we can compare networks between populations where selection has differed for cooperation. And a theme of this meeting, I guess, is kind of the temporal analysis, so actually looking at how individuals change their ties through time in relation to events, sort of cooperation and defection. Thinking about sexual selection, well, we need to have really rich, complete data on who mates with whom, where and when, in order to have that, in order to be able to draw those sexual, construct those sexual networks. And the same is true for collective behavior. We really need to know about the position of individuals in space and time and how that relates to their social network. And again, collecting that data is non-trivial. This Starling work was done using still photographs, and even then, it's still very challenging in order to track individuals, but these individuals are not individually identifiable. So we somehow need to be able to integrate those things together. So for some of these questions, we need to have dense, data-rich social network data. And how may we go about doing that? Well, I think it's been termed reality mining and mining real-world systems through the deployment of sensors or through the deployment of machine-sensed data. I think it's really going to change the way we do behavioural ecology. For species that we can do this, we can't put these tags on guppies, but there might be other ways of collecting data. So we can get beyond kind of traditional survey methods and actually, rather than sampling, have complete information on what's going on in these systems. So I can't believe this was only 2006 looking at the mobile phone, obviously before the time of smartphones, but these authors, Eagle and Penland, were the first to kind of coin this term and think about how this may be used by deploying these phones amongst, I think there were undergraduate students or postgraduate students, and studying their friendship networks and the networks based on physical proximity, and they're able to show that simple proximity actually gives a really good indication of the underlying friendship network. But this data provided constant information on where individuals were in space and time in a way that we just simply wouldn't be able to get by our shoal captures of guppies or photographs. And so there's great excitement in applying this approach to humans. Here's a review paper by Jens, kind of outlining the potential there and some of the approaches. This can include video tracking. So here's if I can find my cursor. There it is, white on a white background. Here we can see there's a number of these being developed. Again, this isn't my work, but a fly walking tracking program. This is tracking Drosophila walking around a plate. But anyone interested in kind of quantifying social interaction should be able to see the potential here. It doesn't come without its problems. Having talked to people that's worked with this type of software, individuals might cross over, and actually there's quite a lot of cleaning to do. If we're not fortunate enough to be able to put 20 or 30 animals on a plate in the lab, then we have to think of other ways of doing this, and we might deploy technology on the animals. So proximity loggers here that work on a radio signal have been very common and widely used. Or deploy GPS tags, for example, on animals and give us spatial position, real-time sampling. I think this is great, but just like we had to ask questions before about are these methods really appropriate, we need to think about, is this data really telling us what we think it is? And I had to learn a rather hard way when answering that question. So we've been working with a project on dairy cattle, which is interesting because they have this very dynamic environment where cows join and leave groups due to management decisions of farmers, and there's concerns about social stress and various other things going on. So we decided we could do a really nice study by deploying proximity loggers on cattle that would record their social interactions. And these are well established, so they've been used to look at disease transmission, social affiliations, predator behavior, population dynamics, etc. And they simply work by a collar on this case a cow, and when these collars come into contact, they record and log that event. So let's just check the data's telling us what we think it's telling us before we deploy all 120 of them. Let's put 20 on the cows. And we got quite a shock. So here's a figure that's based or derived from a non-symmetric association matrix, whereby it's looking at collar 1 logging collar 2 and collar 2 logging collar 1, and just taking the sum of time those collars have been logged in contact, but then looking at the bias, so looking at the tendency for collar 1 to oversample against all of the collars, or collar 5 to undersample against all of the collars. And this is huge. This is 23% on average. Collar 1 had a positive bias in the matrix, and around 15% on average, collar 5 had a negative bias. So all of a sudden we've put something on our animals that we think is going to give us this real-time, accurate data, and actually it's just introduced a sampling bias. It's no surprise when you draw that network. Collar 1, cow with collar 1 is the central node that has lots of interactions with all other individuals, and poor little 5 is on her own, very few friends out there on the periphery of the herd. It's simply an artifact of this technology that we've deployed on the animals. Fortunately, we were able to come up with a correction for this, and so we can still work with this, work with these collars, because we're able to show that this is a stable property of the collar. So it's very consistent through time. It's something we can measure, and so something we can correct. But I think it's an important lesson about actually just checking and calibrating that these things are really telling us what we think they are. But not to kind of dampen the message, once we get over that, it's really exciting. So for this cow project, we've got 12 million contact points of cows over 4 months. So really rich, incredible data, and machine sense data on their milk yield, their infections. So we know 3 times a day what the milk yield of each cow was over a 4-month period, and we can relate that back to their social behavior. These big data sets generate new problems and new challenges. I mean, all of a sudden we're confronted with 12 million events rather than 7 events that we had for the first example with the guppies. And we really need to think carefully about calibrating and validating methods before we get really excited and carried away with them. Just to kind of include this, because I do think it's something that would be good to talk about, comparisons are difficult. So comparing networks consisting of the same individuals through time, that was the guppy example, that's easy, as long as the sampling is standardized. Comparing networks in the lab where we have different individuals but the same number, the same sampling is easy and very powerful. But there's a real problem in comparing networks when the individuals differ, the sampling differs, the species differs, and it'd be great if we can find a way of doing this, but currently I'm still scratching my head as to thinking about how we can really compare networks, and I think there's some really difficult challenges there ahead in terms of the influence that sampling and the size, the number, the density can have on virtually all of the measures that we can derive from that data. So just kind of to finish, what about the future? So going back to this graph, I think we can continue that steep upward trajectory, and we've still got to kind of tap into the real potential of this approach. one way we can do this, I think, is by embracing new technology, but we need to do that carefully and in a considerate way. I haven't really talked about temporal dynamics because Dave's about to give us a good insight into that, but I think this is key, rather than taking aggregated data, to think about the dynamics that underpin that data, but in relation to the question. So cooperation is a classic one, diffusion of information, sexual selection, in terms of, doesn't just matter who's mated with whom, but when they're mated with them is going to be important. Experiments on systems we can run experiments on, I think, are underused but really powerful. And this is why I'm really excited to be here. I really do believe that the future is about collaborating, but we need people from all disciplines to get engaged with this, from working on technology to deploy on animals to sense, right through to kind of statisticians, mathematicians, computer scientists. So yeah, I'm really thankful to be here and excited to be here, and yeah, thank you, Dave, for inviting me, giving me the talk topic. So I think that's the last one. Okay.